All right, so we're going to look at some proteins that are embedded in membranes which help with moving substances across the membrane. So, to facilitate means to help or to make easier. And in this diagram, only small non-charged molecules have free passage through the membrane itself. Everything else would need assistance. Water itself, again, because it's so small, even though it's polar, some of it will sneak through on its own, but an aquaporin channel will help it move much more quickly across the membrane. Now, when we get into membrane polarization, there is a set voltage that exists within inside membranes because of ions and charged particles. When we look at this from a human perspective, a lot of times the easiest example is the nervous system. So to send a message from, let's say, your skin or from a sensory receptor, to conduct that message has to do with membrane polarization. It goes from the sensory neuron to the interneurons up in your spinal cord and brain, and back out through your motor neurons. All of this passage of information happening electrically has everything to do with membrane polarization. So when we look at ions, again, they're charged. So this charged substance cannot go through the membrane on its own because the membrane is impermeable to charged ions because of the lipid tails and the hydrophobic area here. So we need voltage-gated ion channels to help. Now we're going to look at sodium and potassium ions and sodium and potassium gated channels. So they're not just open on their own. There has to be a type of stimulus that's going to trigger that opening. And some of them can be ligand gated, but we're mostly looking at the voltage gated ion channels. Now the resting potential, the internal membrane of the axon or the sending arm of a neuron is going to be a -70. So it is hanging out at a -70, even though we're talking about potassium and sodium ions, which are both positive inside. There are these large protein anions which keep this at a -70. However, that can change. So some type of a stimulus can trigger the initial opening of sodium ion channels, and these positive ions heading into the internal part of the membrane can shift the resting potential from -70 up to -55. When that threshold reaches, a lot of the sodium ion gates fly open and sodium ions are rushing in. This creates depolarization where the voltage on the inside of the membrane goes all the way up to +35. That begins our action potential. So we're starting to send this message, but to send the next impulse, we need to repolarize the internal membrane. And to repolarize and bring it back to -70, we have to get rid of some positive ions here. And this +35 voltage is going to close the sodium channels and open the potassium channels. And there'll be a little delay, but when these potassium channels open, because of concentration gradients, the potassium ions are going to head out. As they head out, again, that internal membrane potential goes to -70. Now, -70 is our resting potential. However, we just exchanged a lot of sodium and potassium to other sides of the membrane. So we need to establish the resting concentrations of sodium and potassium. So we do that with the sodium-potassium pump. Now this is how it's often written. And when we think of ACE, we think of enzymes. We are going to change the molecular structure of ATP as this happens. But the sodium-potassium pump is a carrier protein that's embedded in the membrane. And we call it a pump because it's using ATP. And it will also, in addition to establishing resting concentration, it will maintain that polarization of -70 until the next stimulus is received and the sodium channels open once again. So Initially, this is our carrier protein, the sodium-potassium pump. It is not a sodium voltage-gated sodium ion channel because it's going to hold these and change in conformation and change in shape in order to get them to the other side. Now, what's unique about the sodium-potassium pump is it's going to move both sodium and potassium ions across the membrane. In this shape right here, it's open to the sodium ions here. Now the sodium ions will bind here, and when they bind on, in order to get them to the other side, ATP is broken down to ADP, and that third phosphate is bound onto the carrier protein. When you chemically change this location, it changes the shape of the protein, which then sends the sodium ions to the other side. When it's on the other side, now it is receptive to the potassium ions. When the potassium ions bind onto this side, in order to get them to the other side, we have to change the shape of the carrier protein again. And that is done as the phosphate group is released. And that's going to change the shape so that the potassium is now open to this side. And again, this process can continue on and on and on until resting concentrations are established and polarization will then be maintained. So, through facilitated diffusion, we have aquaporins helping pass water across membranes. We have voltage-gated ion channels, which help with polarizing membranes. And the sodium-potassium pump establishes concentration gradients and maintains membrane potentials.